All right. So today's lecture, we're going to continue with tumor profiling. In the last lecture, we had Dr. Natty Getz, who gave a really comprehensive lecture on how to do tumor mutation profiles from high-throughput sequencing, whether it's just sequencing the exons of genes or sequencing the whole genome and using mutation costs to figure out, what are the mutation patterns, what they mean, and whether something are driver genes. So actually within the big TCGA tumor consortium, they profiled over 10,000 tumors, and DNA sequencing is only one of the platforms. That's the very top you can see from mutation. From the DNA sequencing, you could look at copy number, but most of the copy number variation profiles are actually done using microarrays to look at copy number changes. But in addition to that, they also did gene expression. They also did DNA methylation, microRNA expression, and RPPA is a proteomics approach. And then they also have some very basic clinical annotations. For example, patient age, gender, the tumor stage. Some of them have basic treatment and also survival, at least in 5-6 years. And so these information are all available. And so this is a really comprehensive resource for us to look at tumor in a holistic view, not just from the mutation side. And so by this time, a TCGA project has already finished, and there are 33 different tumor types and over close to about 11,000 tumors that are profiled. And today we're going to talk about, in addition to the mutation cause, what are other interesting things you can look at in terms of these tumor profiles. For the first two modules of this course, we talk a lot about gene expression. I think the importance of tumor mutation is that they do influence gene expression. And so with TCGA, they also did tumor expression. In the very early days, they used just microarrays. When the TCGA project was started, it was like over a decade ago. Initially, they did microarray, and then they did a little shallower sequencing, but at the end, they just did deeper sequencing of all the samples. And so now we have good RNA-seq data for all of the tumors. In terms of data analysis, you can imagine you can look at the differential expression. This is just one example. Each column is one cancer type. The red is the tumor, the blue is the normal, and each dot is one sample. And if you just look at all the samples together, this is just for one gene expression, you can see roughly how high is this gene expressed in the different tumor types, whether it's higher expressed in the tumor compared to the normal, and whether it's a difference in the tumor stage, for example, from the primary tumor to the metastatic tumor, and so on. Right? So that's the very basic analysis. You can imagine after you do differential gene expression, you can also run gene set enrichment analysis and things like that. So with many samples available, you can also look at the gene modules. This is when you have many different samples, you can do this. You can't just do this with just a tumor normal pair. So this is called WGCNA. It's a weighted gene co-expression network analysis. You can imagine if you have, say, one tumor type, let's just think about like luminal breast cancer in hundreds of samples at the same time, you profiled many patients, then you could look at genes that are correlated in many samples. And so in this case, they could see this very big cluster of genes that are correlated, and then there are smaller other gene groups that are correlated, and each might be enriched in a different pathway or some different functions, and you can use this to identify robust gene modules that are co-expressed in the many tumor samples together. Usually you do this, cancer type by cancer type. Okay, so this, and then you can ask whether those group of genes are co-regulated in some similar ways. You can imagine looking at ChIP-seq data or based on the genes in this module, you can run algorithms such as LISA to see what are the transcription factors that are really important in this type of gene modules. Okay. Another thing that was very useful is pathway enrichment. We mentioned that if you have differential gene expression, especially between tumor and normal, you can run gene site enrichment. Earlier in this course, in many homeworks, you have tried to run either David or gene site enrichment. But sometimes if you have many samples, such as a cohort like TCGA, say in a lung adenocarcinoma cohort, you might have 300, 400 samples, and you want to probably see how often this pathway are differentially expressed in the different samples. And so for that, the Broad Institute now have this single sample GSEA. Basically, you have to first decide what type of pathways you are interested in. Remember, for gene site enrichment, there are probably tens of thousands or maybe hundreds of thousands of different pathways. You can focus on looking, for example, only at metabolism-related pathways or gene sets. You can also look at immune-related pathways. So you predefine the set of signatures you want to look at, and then the single sample GSEA, they have kind of some reference. You know, they probably look at many gene expression data together to establish a baseline. And then based on the expression profile you observed in every single sample, it can be compared with this baseline, and then it can show you the level of gene site enrichment of this one sample in this one signature. So in this case, each column is one sample. You can see here is patient 12, patient 11, or patient. So these are patients. And then each row is one pathway or signature that's being displayed in here. So you can evaluate for a set pathways how much it is present or absent, or how strong the signature is present in these individual tumor samples. So that's kind of a way for you to look at different samples to see how robust a particular signature is compared to the tumor versus normal, or within different tumor samples in the same cancer type. Okay, so that's another way you can look at gene expression. So that's a little bit, you know, it kind of uses what we have learned earlier in the semester, but you can see there are many other tools that are developed to make these analysis even more meaningful when you really have many samples available. Another thing that people are very interested in is doing tumor subtyping. These are very often, you know, in the early days, these are mostly done in pathology. So you can imagine this is in breast cancer. Many years ago, breast cancer in pathologists know that after you have a tumor slide, it's good to stain for 3 different markers. One is estrogen receptor, one is progesterone receptor, and one is HER2. So these 3 receptors. And at that time, they know that if the patient has a estrogen receptor or progesterone receptor positive tumor, they have usually better outcome of the patient, and they should be treated with something. And with early days with HER2 positive breast cancer, in early days, these patients have very bad outcome until a HER2 antibody therapeutic antibody was developed by Genentech. It's called Herceptin. And patients with the HER2 amplified tumors, once it's treated with Herceptin, has much better survival. Not as good as the ER positive tumors, but it's still much better than without the Herceptin drug. And then there's another type, which in the early days, people knew if the patient it's called the triple negative. They are estrogen receptor negative, progesterone receptor negative, or HER2 negative. And so if these are triple negative breast cancers, they usually have much worse survival. And in 2001, this is even before RNA sequencing was available. It was using microarrays to look at expression of thousands of genes. People can already start dividing the tumors into subtypes, and you can see that in addition to the ER positive, HER2 positive, or triple negative as 3 subtypes, they were able to use just basic hierarchical clustering to divide the breast cancer into more subtypes. So for example, there's a Lumina A type, which is fairly frequent. By the way, on the right side, you can see how often these type of subtypes really occur in the population in the early diagnosis. For example, in the primary tumors, about 40% of those are Lumina A subtypes. You can see here, these are also a bigger population. And then a smaller one is called the Lumina B. It's 26%. It's still actually, I would say, quite abundant. And so between these two, you probably are talking about 60 to 70% of all the breast cancer cases, right? And then there is, there's also, at that time there was Lumino C and a normal-like. This is, normal-like is the 13% in here. And then there's HER2 positive, which is about 10%. And then there's 10% basal-like. These are the traditionally what people call triple negative breast cancer. And these are usually pretty bad. And so based on years of people using either microarrays or RNA-seq, they can do the clustering and correlate with patient survival or pathology. So scientists developed this panel called PAM50, basically using 50 genes as markers to decide whether a particular tumor belongs to Lumino A, Lumino B, HER2, basal-like, or normal-like tumors. By the way, this normal is not real normal, it's normal-like. They are still kind of a breast cancer type, but they usually have pretty good outcome. For example, if you look at the metastatic tumors, so these are, patients, either they had a recurrence or at the time of diagnosis, the tumor has already metastasized. And so if you look at their subtyping using these 50 genes, and so by the way, the 50 genes are now FDA approved subtypes. Basically, when the patient goes to the clinic, they can use a simple assay just to look at the expression of these 50 genes to decide what subtype they belong to. And this will be covered by the medical insurance, because depending on what subtypes they have, the patient will be treated with a different type of drugs, and they will also know that they will have different type of outcome. And so you can see here the metastatic tumor compared to the primary tumor. You can see the primary tumor has much more Lumino A, a little bit less of Lumino B, but fewer HER2 or basal-like. Whereas if you look at the metastatic tumors, there are a lot fewer Lumina A subtype, because the Lumina A is a good, kind of slightly better outcome tumor. And probably the best is normal-like. Because they are normal-like, they kind of grow slower, they're less likely to metastasize. It's probably in this order. Normal-like is the best type, and then Lumino A, Lumino B. HER2, I guess, even though they do appear a lot in the metastatic tumors, you can treat the patient now with a drug, with a therapeutic antibody drug, which is a good sign. But then with basal or these triple negative breast cancers, the treatment options are much more limited, and the patient will have overall worse survival. So you can see the subtyping are very important. The patient will be treated differently, and they will have a different outcome. This is in the early days when people were only using gene expression. But as we mentioned with TCGA—
I have a question.
Yes.
Yeah. When you say from metastatic tissue, do you mean like this is DNA? This is like DNA from the actual like metastatic tumor that like went to the lung or went to the liver, like went to the lung or went to the brain, or is it from patients that just have a metastasis in general?
I don't know specifically for this type, but for example, sometimes when the patient have a surgery, the doctor might already discover that the tumor has metastasized to the lymph node.
Yeah.
Then they could resect those tumors. And when they do this, it's not just DNA. Actually, for the subtype, it's done on the RNA, not on the DNA.
Right. Yeah. Yeah.
Yeah.
Yeah. I was just wondering, is this like looking at the tissue from the lymph nodes, or is it looking still at like just a breast tissue from someone who has metastatic disease?
It really depends on the sample availability. For example, for melanoma samples, because it's on the skin, usually you can easily resect the metastatic tumor. But for example, if it's breast cancer metastasized to the liver, then patients usually don't want to be operated on. Even for biopsy, it carries risk for the patient.
Okay.
If it's from surgery and the doctor already see a lymph node metastasis, it's easy to just take it out, right?
Okay.
So doctors usually do it. And so it really depends. So in Tuesday's lecture, Dr. Getz mentioned that nowadays in the cancer research community, they're trying to get really early early cancer lesions. But you need to have early diagnosis as well, which is tricky. But at the same time, you also have we are always interested in metastatic tumors because primary tumors don't usually kill patients. It's usually metastatic tumors. The difficulty there is very often when the tumor are metastasized, especially for a distal metastasize, patients usually don't want to do surgery because you see one metastatic site, metastatic tumors usually go to tissues that have a lot of blood, lung, liver, bones, and brains. And those places, it's hard, first of all, it's harder to operate on, and it carries risk. And just because you don't see other potential metastatic sites doesn't mean that they don't have those. And so just operate on one metastatic site would not help the other sites.
Yeah.
Right.
Right,
So,
thank you.
yeah, the only exception for that is a brain metastasize. For example, if somebody have a lung cancer that's metastasized to the lung, to the brain, sometimes without operation, it could influence the patient breathing. It could influence the patient for balance, and depending on where that is in the brain, right? A patient may not be able to walk, or they can't see anymore. And so most of the time, patients do not want their metastatic tumors to be operated on. The exception is when it's metastasized to the brain. And so actually at MGH, there are surgeons, brain cancer surgeon like Dr. Priscilla Bricianos. They were able to operate on the brain because they have to take it out for the patient normal brain to function normally. Because if it's metastasized to the other organs, the organ can grow, but when it's in the brain, the skull limits the total size. And so the tumor can squeeze important other brain tissues, so they can't work well, right? So they can get the metastatic tissues to do more profiling, and they are making really fantastic discoveries. Okay, so
I also have a question, sorry.
Yeah, yes, yeah.
So I can see from this slide that tumor is really heterogeneous. And then, for example, when we are doing experiments, we normally would take a cancer cell line to try to model the like human cancer type. So if we are using, like, say, a cancer cell line from Cancer Cell Line Encyclopedia, and is it sufficient if we just try to plot the correlation between the, for example, kidney cancer cell line with all those kidney cancer subtypes in the TCGA in terms of RNA-seq, which is gene expression, to show like which human cancer type does this kidney cancer cell line best model? Or do we also have to consider like protein expression, mutation, or DNA copy number changes, and combine all those 4 indicators along with RNA-seq, which is gene expression, to determine which human cancer type does this specific cancer cell line can best model?
So the question is whether cancer cell lines are representative. I guess we have 2 questions. One is just because you get a cell line from a breast cancer patient, then you can propagate this. For example, the Broad Institute has a really big cell line encyclopedia. They have about 1,400 cancer cell lines now. How well does this cell line capture what happens in the primary tumor? I would say it's only one representation. I will show you later on with single cell tumor profiling. It's informative, but it's not everything. This is one thing. In addition, what is useful information to profile? For example, that's really a matter of cost, right? The Broad Institute initially, when you have 1,400 cell lines, you do, they did SNP arrays, they did RNA-seq, they did like whole exome sequencing to look at the mutations. Recently, they just published a proteomics data for those cell lines, and they're doing CRISPR screen. So every assay to do this consistently across many cell lines, there's cost. And so biologically, the cell line represents some of these tumors, but maybe not completely. So for example, even with breast cancer, you can use the PAM50 to decide whether the cell line belongs to a luminal A or basal. But even after that, it's only one type of basal out of maybe many different basal tumors, right? Is it representative enough? Probably not. But also, what type of profiling are informative? I think the more the better, but it's just a matter of cost. Can we afford to do so many, or at least abroad, can they afford to do so many?
Thank you so much.
Sure. And so, for example, with TCGA, most of the samples have also gone through epigenetic profiling. At that time, they did DNA methylation. In the early days, it was a different DNA methylation array. Later on, all of the samples are profiled with this Illumina human methylation 450 array. Very few samples were profiled with whole genome sequencing. You can see this 450 array. It has this many number of CPG sites, and it covers, I think, all the genes promoters, especially if it's CPG-rich CPG island promoter. And compared to the very early day arrays, that the newer ones also cover some on the gene body, on the enhancers, some in intergenic regions. And this really has been done. All the tumor types, I would say most of the samples in those tumor types have DNA methylation readout. Recently, there are also efforts doing whole genome bisulfite sequencing. Of course, this is very expensive because we have to sequence the genome many times over, right, in order to read out this. So this is only done on very limited numbers. And so what an analysis you could do early on very easily, we can check is, you could look at whether the promoter, if the promoter of the gene has a CPG island, you can see whether that CPG island is methylated. And if it is methylated, we will know that in this tumor, this gene is silent, it's not expressed. And we can also see if the promoter is not methylated and also the gene body is methylated, that actually indicates this gene is highly expressed. And recently, there are even more work looking at DNA methylation on the enhancers. And those can be used to explore what transcription factors might be binding. Of course, what we know is that if it's methylated, it's probably not binding. But if it's unmethylated, it doesn't mean that it is binding. So it gives you some information, but not all the information. When TCGA was available, actually the community discussed whether we could do ChIP-seq or some chromatin accessibility assays, but at that time the technology was not ready, so it wasn't done. Recently, Howard Chang's lab at Stanford generated the ATAC-seq data of about 400 TCGA tumors. It's not everything. They picked representative tumors from each cancer type to do this type of profiling. And so with ATAC-seq data, now we can look at transcription factor binding in many different cancer types much better now. So that's but it's not done as systematically as DNA methylation or RNA sequencing or mutation analysis. Another profiling technique is proteomics. there are actually many different type of proteomics. One is mass spec, but those, I think depending on how many proteins you want to detect, the cost is still quite different. What has been done for most of TCGA samples are these protein microarrays. So by the way, these are two figures I found from Google Image. They both kind of explain what this reverse phase protein microarrays are. Basically, when you have a tumor sample, and so in this case, each column is one tumor, and you kind of take a small slice and you put it on the array, and they put 3 replicates in here. You see, these are replicates. And you can also do the dilution initially. So you get the protein at a low level, at high level. And so this is one sample. And then this is another tumor, this is another tumor, or another sample basically. And so there's a dilution, there's also replicates. And then with this, they have to generate many such arrays, because you can see each tumor, you can take very thin slices of this to put on this protein microarray. And then for each array, you can put in many tumors in here. See, each block is one tumor. And then if we're interested in testing the expression of one gene, for example, KRAS, I want to see how much KRAS is expressed or protein level is available on each tumor, you just put an antibody against that protein. And if the tumor is expressing high level of that protein, the antibody will bind strongly. But in order to really detect that, later on we use another fluorescently labeled protein which will recognize other antibodies to bind to this. And then when you shine lights, those fluorescent antibody can give out lights of different colors. And so the really bright ones are the tumor samples that have high levels of the protein you're interested in. And so you can imagine for the same similar slides, you can either just wash them off and then test on another antibody, or you can take a different tumor slides and try a different primary, like first antibody. So the first antibody detect the target you are interested in. The second antibody only help you detect how much the first antibody is on the array. And so at the end, you could know each tumor how much they have for this type of, for any protein you have a good antibody for. And so for TCGA, they have, I think, something like 300 different antibodies on proteins that they know are potentially interesting or useful to know. And so they also profiled thousands of samples, and that data is also publicly available. And so now with all the data available, you wonder whether you can cluster them together. And so for that, initially when people were trying to cluster based on mutations, you can imagine if you look at all the tumors with HER2 mutation, they don't always cluster well as what you get from cluster based on RNA. But if you cluster based on microRNA, it also gives you a different clustering. If you cluster based on DNA methylation, it also gives you a different clustering approach. And so at the end, recently people are using consensus clustering. You can imagine, remember consensus clustering is initially you can just take a subset of those genes and also take a subset of the tumors, and then you try to do k-means clustering, and then you ask, do 2 tumors get clustered together, co-occurrence matrix, whether they belong to the same cluster, and if so, you write a 1 in there. Next time you sample another group of genes and you sample another group of tumors, you do clustering again, and you do this co-occurrence again. And if 2 tumors are again within the same cluster, you write to your co-occurrence matrix again. But you can also, so initially, people use this to look at what genes or what tumors are robustly co-clustered in the same group. But you can imagine you can use the same approach to combine different profiling techniques So for example, sometimes you can do clustering with just whole exome data. Sometimes you can do clustering with RNA-seq data, with microRNA data, with DNA methylation data. And then you just ask how many of those tumors are always together, regardless of what platform you have. And this way, at the end, you can get, hopefully, better clustering result. So you can see here from the top, this is from the RPPA. This generates some subtype. This is breast cancer. You can look at the ER and PR status. You can see actually these two are kind of correlated. HER2 is a distinct cluster. You can also look at pathology, how well they looking at, let's say, tumor size, how well you stain the tumor and other, depending on what markers you use. And this is using mRNA expression. This is using mutation status, and try to look at whether you can cluster them together. And so, with more data, potentially you can cluster the results even tighter. But then, of course, there are maybe some tumor samples, they're just oddball, look very different from others, right? And so, the reason we are very, very interested in knowing about the subtype is because depending on the subtypes, the patient will be treated differently. They will also have potentially different survival. Okay, so that's why people are interested. There are many, many resources available on tumors, especially on TCGA. So for example, with TCGA, the Broad has a Firehose, and University of Chicago has this GDC, Genomics Data Common, has a lot of the tumor-related resources. And actually from the GDC, you can download the raw data from the TCGA, and as well as process the data. The CBio portal is something that I think homework 6, you will play with the data there. This has all the process data, but it has really user-friendly visualization tools that you can use. Yeah, UC Santa Cruz has some resource that also collected the TCGA data, allowing you to do, you can access both the raw and process data, but also there are APIs allowing you to do data analysis on the server. And University of Michigan has Oncomine, which has many other non-TCGA cohorts. They are collected from individual studies you can look at. And also there are, Sanger has this Cosmic, which has all the a catalog of all the mutations in cancer and annotations for those, which are very, very useful. Okay, questions?
Yeah, I have a few questions.
Uh-huh.
So is there any resources for like mouse bulk RNA-seq data?
That's a good question.
Like mouse
That I'm not aware of. It's a really good question. Yeah, because you can imagine the tumor in the mouse, there are many different possibilities, whether it's a wild type mouse, whether it's a genetically induced tumor, or it's a spontaneous tumor, or whether it's a human tumor that's grown in the mouse, whether the mouse have immune systems. So it's very complicated. I'm not aware of a good database with those collections, but let me know if you know of any.
Okay. And also another question is, for your previous slide that you introduced the TCGA cluster, which is a multi-omic, like integrative techniques. So is there any like algorithm that you can detect, for example, each tumor subtype specific upregulated gene cluster, like in using the iCluster?
Yeah, so after you divide the genes the samples into clusters, then you can just do regular differential gene expression.
Do regular differential gene expression. Yeah. But for example, if you cluster all those cancer panels in TCGA into, for example, 18 clusters, and if you're going to detect what are the gene sets that are uniquely differentially expressed in each cluster, then you have to do pairwise test, which you're going to do like choose 2 from 18, which will be like 153.
It's a good question. So within TCGA, they don't try to really compare different cancer types. Okay. The potential reason is there are too many differences between, say, a brain cancer versus a lung cancer. Besides, with TCGA, all the brain cancers are profiled at one site. how the lung cancers are profiled at the different sites. And so some of those differences could also be because of batch effect. These are different sequencing center. And so they don't try to do that. Usually the subtype is within the cancer type. For example, breast cancer. Over 1,000 breast tumors are all profiled at the same place. And when they do subtype, with breast tumor, usually people just do like 4 to 5. For that, you can, you can do differential expression analysis.
Okay. And another small question, can you go back to your slides on SSGC?
Here. Yes.
Yeah. So is this like the previous one? Like yeah. So is this the co-expressed network, the module, gene modules? If you're going to annotate with like the annotation package you loaded, it will lead to like the GC is the same thing as GC if you just annotate those co-expressed gene modules.
You mean GC, you mean differential expression?
Yeah, so I mean, I think, correct me if I'm wrong. So if, from my understanding, I mean, like GC is just you annotate from the annotate package you're loading and to those co-expression or co-expression gene modules, right?
Yeah, so it depends on what you have. For example, you can imagine with the broad CCLE 1400 breast cancer cell lines. You could use it to look at, so I would say for tumor samples, people have been using WGCNA to look at gene modules, but if you go to 2 lectures from now, thinking about the tumor immunology, you will know that this type of analysis, depending on the tumor microenvironment, may or may not give you all the correct biological meaningful clusters, but also depends on what you feed the algorithm. You can imagine if you give it all 1,000 breast cancer versus just all the luminal A breast tumors, it will give you, how kind of a coarse grain versus fine grained clusters, right? Because if you just give it all of the breast cancer, yeah, it will give you pretty much the subtypes. But if you only give it the tumors with one subtype, it will probably give you more expression correlated genes that are related to that subtype.
Okay, thank you. So
but I can tell you later on, this is quite confounded by some of the immune genes in there.
Okay.
That's related to 2 lectures from now.
Okay, thank you.
Okay. All right. So I'm gonna stop recording. Yeah, so in the TCGA project, after the scientists profiled many different tumors, usually the consortium will get together to decide how many subtypes do we have. In each major tumor types, they have to decide how many subtypes, right? Do we go with, you can imagine this is like a K-means clustering. Do we go with K equals to 3 or 5 or 25, right? So the question is, do we see a significant difference in the mutations or the expression or the treatment decisions? I think the most important thing is whether the patient will have a different survival and whether they should be treated with something different. And that will determine how they decide how many subtypes. And so looking at that, understanding the survival of the patient in each subtype would be very, very important. And so for that, we'll have a very, very short survival analysis. By the way, survival analysis can be a whole semester course. We're just giving you like a 10 minutes quick overview of survival analysis. And for example, you can use survival analysis to answer a number of interesting questions, such as, do patients receiving a particular type of treatment live longer? Or you can ask, are smokers more likely to have cancer recurrence or, cancer, not recurrence, like a cancer incidence? So patients with this particular subtype, are they more likely to metastasize, right? And so for that, anytime you deal with a real patient in the in the survival analysis, there's a very realistic thing called censored data. The value of the measurement or observation could be only partially known. For example, some patient has left the study, or the study has concluded the patient hasn't died, but doesn't mean that he will not die after the study finished. And somebody, for example, we do also see cases where you call the family and they don't answer the phone anymore. They kind of, when they leave the study, it could be because of few things. They can't tolerate the drug anymore, or they are just too sick, or they don't want to do it. They're like, okay, they just say, oh, I found an alternative medicine. They talk to a shaman or whatever. So it's many reasons when people could just leave the study. And so, for example, we are showing day here, but in a real clinical study, this could be months, could be weeks, right? So initially, you might have 4 people under study, and then usually in these studies, you have a time interval, right? You look at every week or every day or every year, and you ask, okay, so say at this second day, 1 patient died, you still have 4 patients in here, right? So the probability of death is 25%. The probability of survival is 75%. But then, say in the 3rd day, no patients died, but then 1 patient that just they either moved away or they decided they don't want to get this treatment or whatever, you just can't follow them anymore. And so you don't observe anything yet. And then the next one, you can see here, it's this patient died. And that's basically 2 patients, 1 died. That's a 50% death rate. But if you look at your total, like the survival, it's kind of at this 75% divided by half, right? Because 1 patient kind of left here halfway. And then at the last point, there's say another patient who left the study. And this, if they leave the study, we don't know. We just assume that they're still alive, right? So there's still 38% survival. And so you can see if you have a real study like this, so this is your total number of patients that are still in the study. This is say either days or weeks in the study. And this is the event. This event could be a death event, right? So in 0 day, you have 21 patients going into the trial. In the first time point, 2 patients died. And you can calculate, 19 divided by 21, this is 90%. But yeah, so here is, if you don't consider anybody, if you consider, you don't worry about the censored data, you can see here basically, then 2 people died, then it's 81%. And then you have 17 people left, 1 person died. And so you can calculate basically this survival probability. It's the number of patients that passed a certain time point, and then divided by your total patient in the very beginning, right? The reality is that a lot of times, in addition to the people who potentially died, there are also other patients who for some reason just left the study because they you just can't keep track of them anymore. And so in survival, if we consider censored data, in this case, initially, yes, we did have 21 patients. At this time point, we know that 3 patients died. And so you can calculate the probability of the survival in here. But then in the next point, you don't have 18 patients left because 1 guy is gone. Somehow they dropped this from the study. And so the denominator is no longer 18, it's only 17. It's the available number of patients who are continuing on with the study. And then 1 died, and so the survival probability is actually 0.86, not 16 divided by 18, right? And then say another patient left. So you can see here the difference is that denominator is always just the number of patients who are still left in the study. And that's kind of important. When you calculate this final survival curve, what you are showing is the this probability over time, you know, and you can compare it with two groups. For example, if somebody goes through one treatment versus the other treatment, there you can't have people leaving the study. So in this case, every time you have a drop, you know, this probability go down, is because there is a patient who, for example, either their tumor relapsed or the patient died. It depends on what you are really looking at as the event. But anytime you see this little tick in here, you see here the little bar, that means a patient left the study. You just don't have information for that patient anymore. And so those are, you know, just the natural events which happens in a lot of these clinical studies. But when you calculate the patients, you only use the available number of patients to calculate the probability at each time point. And so this is a very standard Kaplan-Meier curve. And so you are comparing the two groups and you're asking, are they significantly different? If you don't know the statistics, you can just, you know, see you want these two curves to be as different as possible. If they are really far apart, you have a very significant p-value. So KM curve, easy to read, right? But if you want to see how some of the probabilities are calculated for survival, there are two types of tests you could do. One is a log rank test. So you can see here, each row here is an event, either it's like a day or week, you know, the time point that you are looking at. And then, let's see. This is the number of failures. Failure means either the patient died, or in this case, or it could be relapse, or you know, whatever event you want to capture. And this is the how many patients are still in the set in the two groups, right? So, but then, so you can imagine. Let's see. You are trying to compare the two groups, and you are asking how many events. So initially, you have two groups, each have 21 patients, and then at the first time point, let's just say 2 people died in this group. And in the second day, 2 people died again in this group. The third day, another 2 people died in this group, right? So you can see in this situation, it's probably more events happening to the second group. As the first group, you know, occasionally there are patients dying, but it seems like a lot more patients are dying from the second group. We're trying to ask whether those type of death events or whatever the events, failure events, happens evenly in the two groups, or they're happening more towards one group from the other, right? And so at every point, you are calculating the expected number of events. For example, because we know there are 2 patients died, so out of the 42 patients, because this group only have half of those patients, you are expecting the death events to also be evenly split, right? So there are 2 total deaths, and because this group have only 21 patients, you expect to have half of it. That's your expected. But the observed is that there is nothing in this group. The observed for here is the 2 patients all coming from one group, right? And then you look at the next group. Again, there are 2 death events, and you're calculating how many total patients are still available and how many are from that first group. And so you can see here, in this calculation, the denominator here are what are the total number of patients that are still available at this time point, and also how many patients are available in my group. You know, in this group is 12 out of 18. This is 6 out of 18. So it's already censored data. What are the available data you are using to calculate this? And so because at every time point you can calculate the observed versus the expected, right? So this is asking, what is your observed over expected? And every time point you can calculate this. At the end, you're asking, is the observed versus expected significantly different. And so for the log rank test, what we are expecting is called proportion in the risk set. You are hoping that the risk should be occurring with the null hypothesis is that the risk for this failure event, whether it's death or recurrence or whatever, is proportional to the sample size that's available at that time point between the two groups. And you're asking how different are they, right? Then you can just use chi-square test to see whether you observed versus expected is significantly different or not. And so normally with log rank text, you are considering two groups and checking whether a treatment is making a difference, or whether patients say whether if they have lung cancer, whether the smokers and non-smokers have different survival, or whether they respond to the drugs better or worse. But sometimes you can also have more variables. You could look at, for example, expression of some genes, or you want to say, in addition to, so for example, we could consider one variable as patient age. You are wondering whether the patient age make a difference. You are wondering at the time of diagnosis, whether the tumor stage make a difference. You are wondering whether the gender makes a difference, whether smoking makes a difference, right? And those variables can also be both discrete, which means it's potentially categorical, or it could be continuous, which is like, say, gene expression value. And so when you have so many different things, you could use a Cox regression to ask, if we look at all of these together, can you see a difference between the different groups? So we have a question. Yes, Jack.
Yeah, sorry, can you go back to the log rank test? Here, can you explain how to exactly calculate this statistic at the bottom? I'm not sure how to do the variance.
How do you, so normally how do you do chi-square test in a 2x2 table? Right? You always have, you have many observations, and you have many expected, because every row is a value.
Yeah,
right?
I'm just kind of lost on like for example, use this example, what kind of number are we plugging in to the formula?
So every row you have expected and observed. Every row you have expected and observed. And you can look at the variance of those, right?
Yeah, it's just can't really remember what kind of formula we should use. Yeah, but
yeah, maybe as I mentioned that, so in general you could look at this in a survival course. I think you can see this explained very clearly.
Yeah. Okay, sure.
Yeah, so basically log rank tests just give you one variable at a time. You ask whether something give you a difference, whereas the Cox regression, they give you many different variables. You can estimate them together, and it can help you estimate a coefficient to try to separate the groups. And it can actually try to help you separate things to see whether some variable can make a big difference. And it can also tell you whether a particular variable is significant or not. For example, if you include many variables in here, if some coefficient is close to 0, you will know that, okay, that variable doesn't really matter at all. But say patient stage is important. If it's stage 1, the patient do better. If it's stage 4, the patient do much worse. You will have a survival result where the coefficient will be quite statistically significant. So when you do this Cox regression model, it will tell you that this variable is significant, the next variable is not so significant. And so you can look at many variables together, and they could be either categorical or continuous, like gene expression levels. Yeah, so that's kind of the different ways you can look at this. Yeah, so let's see. This one is using two groups of genes. So supposedly you have, say, the PAM50 gene expression, right? You can look at 50 genes, and using the Cox regression model, it will help you weigh the patient. So, and then see, looking at the survival, say, this gene really has this effect on survival, that gene has this effect on the survival. And so based on the expression of all the genes, it can help you separate the patient into the low risk versus high risk groups, which can be also very, very useful. So that's how, you know, if you have the differential gene expression from the different groups, you can use that in your Cox regression to see whether the subtypes have different survival, and you know how those genes are associated. By the way, none of these are causal, they are just associations. They don't tell you what this gene is the reason why the patient had better survival. It just says if the expression of these genes are overall like higher, or you know, based on whatever weighting is higher, the patient will have better survival.
Do the ticks still indicate a patient has dropped out?
Yeah, yes,
Okay.
exactly. That's the sensor data.
Okay, so it just seems like, is it, I don't know, it just seems kind of weird that there's a lot of patients that dropped out in the low risk signature. Is that typical, or, I don't, yeah?
It depends on the study, I would say, and also depends on what you mean by the event. It could be a death, it could be a recurrence, it could be whatever reason. Yeah, so it depends on what you're looking for. I don't think it's always the case like this.
I have a question.
Yes.
So how do you derive hazard ratio from the Cox regression?
That I think will probably take a whole course to do. I think it will take a lot longer than this lecture. We can include some videos for, you know, like we can find these similar videos online. Maybe also with Jack, we can find similar videos to show you how to do the log rank test as well.
Yeah. I just figured.
I'll include those videos
If you don't mind, I can answer that, how to calculate hazard ratio. It's just the exponentiate of the beta coefficient you estimate, and you get a hazard ratio for 1 unit increase in that variable.
Okay, thank you, Jack. Yeah, so in the last lecture, Dr. Getz mentioned that they are trying, using the mutation analysis, you can try to find genes that are recurrently mutated. So in general, the hypothesis is that if the tumor is recurrently mutated, or the genes or pathways recurrently mutated, they are likely drivers, right? And in this includes tumor suppressor or oncogene. So tumor suppressor prevents tumor from happening, whereas oncogene, they drive the tumor growth. And very often when you have a tumor that's recurrently mutated, it could be also gain of function mutation or loss of function mutation. For example, very often in the tumors, what you see is a gain of function mutation on the oncogenes, which means that this gene normally can drive cell growth, but it needs some external signal. Usually it's a growth hormone receptor. It needs to have the signal to turn on. But with a mutation, now it can just keep growing without outside hormone signals. And that's a gain of function mutation. A loss of function mutation instead is, if this gene normally is expressed to suppress the cancer from uncontrollable growth, and suddenly this gene is lost, then the cell can keep growing. And so from the mutation, how can you see whether something is either a gain of function mutation or loss of function mutation, right? You could look at some mutation patterns, including the copy number variation patterns. You could also look at their gene expression patterns. Of course, Gary Getz mentioned last lecture that these are all hypothesized. You still need functional experiments to really figure out how this really happened. But of course, some genes can be both tumor suppressors and oncogenes, which are more complicated depending on in which cancer type and exactly where that mutation happens on the gene. It could have opposite effect potentially. And so we talk about in the last lecture this GISTIC score, which basically look at how often a particular, this is looking at copy number change, right? How often this type of copy number change happens, and also the amplitude. Is it 3 copies or 5 copies, or you lose a copy? And then using this, you can look at whether this is a significant difference. So the idea is if something is repeatedly amplified in many tumors, not only they occur frequently, when they occur like 3 times, 5 times, or 10 times, that's usually a very strong indication this is an oncogene, because you amplify it, then the patient will get tumor, right? So the copy number give you information. If you have a copy number loss, then the patient have tumor. That's more likely a tumor suppressor. You can also look at, we mentioned in the last lecture on the specific mutations, and very interesting talk on Tuesday, that mutation rate is not even across the genome. Different locations have different mutation rate. This is potentially related to epigenetics. So basically, if it's open chromatin, they are less likely. So if this region has open chromatin, the genes are highly expressed, they are less likely to be mutated, because very often the gene needs to be expressed. If you have a mutation, it's less likely that's good for the tumor. It's like if you have a car, you randomly take things out. How often is it running better? Most of the time, it will not run very well. Just occasionally, you have a mutation that will make the cancer cell grow much faster. And so most of those mutations will be eliminated because of selection. At the same time, in regions where there are gene expression happening, there are also good DNA damage repair mechanism. They will detect there's something going on. So there is transcription coupled DNA repair mechanism. So highly expressed genes if there are any mutations, they're also likely to be repaired. So it's only in those these regions that are epigenetically silent, not many genes expressed, and like no good repair system, then they get mutated a lot more. And so with this, you can figure out whether mutation happen a lot or not. And when you look at this profile, which we also saw on Tuesday, when interesting observation is that if this is a cancer type that appears in children's, they are less likely to have a lot of mutations. But if it happens in older people, naturally you'll have more mutations. It's so for example, there are patients who take cosmetic surgery to take out some, like the bags on their eyes. And when you take this out to look at their mutations, if it's coming from older individuals, you will see a lot of mutations, even though they don't have cancer, they just have more mutations, because over time, we accumulate a lot more mutations. And also the tumors that face outside, these are tumors that, skin or lung, because we breathe, and also the GI system, those actually face outside. They face the foods, so they deal with outside stimulus all the time. They have overall higher mutations. Whereas the tumor that face inside, such as brain or blood, you can imagine those really, and also say prostate, they don't really face outside. They usually have fewer mutations. And you can look at the mutation pattern very often to see whether this is an oncogene or not. For example, for MYC, if you look at where the mutation happens on this gene, it's kind of scattered. But most of the time, if you look at many different cancer types, most of the time the mutation happens as a copy number amplification. So that just the same gene you just amplify this region, they can have many, many copies. And if you look at the expression, MYC is highly expressed in many, many cancer types. That's usually an indication this is an oncogene, right? Whereas in another case, this is also interesting. You don't see so many copy number gain. But if you look at the mutations, most of these locations don't have mutation, but this one location at G12 locations, there are like 1,300 mutations happening in this exact same location. That's a gain, it's a very clear gain of function mutation. I guess this is where the KRAS needs interaction to be activated, but with the mutation, they don't need the signal. They can always constitutively be on. And so this is clearly a gain of function mutation on oncogene. So this one is kind of more complex. If you look at this mutation, it's kind of all over the place. And also you see a lot of these missense mutation. Green is missense mutation. Missense mutation means that it's an amino acid change. There are also many truncating mutations from the beginning to the end. You can see here, from the beginning to the end, truncating mutations is that you're gonna lose this gene. But also you see more blue, these are deletions. So this is actually a tumor suppressor. You either lose it from copy number or from a particular mutation that basically the rest of the gene are no longer expressed. You have an early stop codon, and so you lose the expression of this gene. That's a clear indication of a tumor suppressor. There are some more complicated patterns. For example, this one, you see P53, it's a very well-known cancer gene. In all the cancers, you don't see a lot of cancer copy number change, but if you look at the mutations, there seem to be some early, you see truncation, a lot of truncations early on, but then there are also missense mutations that happen 400 times. This gene, there's some hypothesis that the wild type gene is a tumor suppressor. So if you completely lose it, the tumor has problem with the DNA damage repair. But interestingly, if it's mutated just in those locations, then it can potentially have oncogenic effect. It can actually promote the tumor growth. Not necessarily a complete gene loss, right? So you can see that's getting a bit complicated. This is another complicated pattern as EZH2. You can see in different cancer types, sometimes it's a point mutation, sometimes red is amplification, and sometimes it's a deletion, and it's complicated. If you look at the expression, it seems to be overexpressed in many cancers, so maybe it's an oncogene, but it could also potentially be a tumor suppressor gene. So you can see it could be quite complicated. If you look at all the tumor mutations from a pan-cancer, all the cancer type together, the blue genes are all the very well-known cancer driver genes, whether it's an oncogene or tumor suppressor. The red ones are kind of novel, but they have clear cancer association, and there are also some really novel ones. The biggest surprise from all these mutation analysis with TCGA or expression analysis is that there are a lot of mutations on chromatin regulators. And because of this, a lot of drugs are now developed to study chromatin regulator changes. So we'll talk very briefly about chromatin regulator mutation as well. Questions?
I have a question.
Mm-hmm.
So has anyone combined the GWAS study as well as ATAC-seq study to look at sort of like SNPs and changes, the SNPs that confer the epigenomic changes and thus, and thereby confer to like inherited variant to the like population level somatic mutation, sort of things like that.
So by the way, in the last module, we talk about a disease, right?
Mm-hmm.
The first lecture, the guest lecture was talking about more germline mutations.
Yeah.
These are, maybe the mom and dad are both okay, and suddenly the child has a spontaneous mutation, or maybe there's just a SNP that happened in 5% of the population, right? That's germline. All of your cells carry the same thing. Here we're talking about somatic mutations. It doesn't happen in all the cells in your body. It only happens in that tumor compared to adjacent normal or blood. They are
Okay.
quite different things, usually.
Mhm.
I would say germline mutations, if it's a very rare disease, it could be a very strong effect. You have this mutation, you get the drug. But if it's kind of appearing in 5% of the population, it's probably not a very strong effect.
Mhm.
Somatic mutation, it depends on, you can see how often they appeared in the tumor. It's probably the more frequent, the stronger the effect.
Mhm.
Yes.
And also, I have a question on the one of the factors that has different expression level in different type of cancer cells. So for that one, because like I suspect this data is from the TCGA, which is like the bulk RNA data. So does anyone like look into like each of these cancer data and do the single cell to see whether there's different expressions because of the specific cell type, and or also because like the different cancer related tumor microenvironment that kind of drives the
everybody is interested in doing single cell. Unfortunately, the TCGA tumors are already frozen. It is not in single cell suspension, so you cannot do single cell RNA-seq on the original TCGA tumor anymore. But but scientists are getting the fresh tumors, getting the single cell suspension. Now they can do either SMART-seq or 10x genomics single cell sequencing. We have a little section at the very end. I'll show you. Okay. So
sorry, also one question.
Yeah.
I just wonder, like, what is the logic behind like a mutation happening many times on one site is a signature of gain of function mutation?
Mm-hmm.
And conversely, why, like, if we have like many mutations with low count,
like this, right? Yeah. So
is a sign of like loss of function.
Yeah. So for example, in this situation, normally this gene has its, say in this case, a normal function in order to say interact with other protein, or it needs a growth signal to grow. But then suddenly you have this one mutation, this gene can be constitutively on. That's recurrent. You really need this position to be on for everything else to happen. It doesn't happen if the mutation happens in another location of this gene. And so when you have very, very focal mutations, very often it's a gain of function mutation. It's a key place, because if this position is a loss of function mutation, then it doesn't matter have to be this space, right? You can just lose it early on. You can have a truncating mutation, you lose the whole gene. That should also work. Right, and so this type of mutation, the IRB, is like you can see, you have copy number loss, the whole gene is gone, or you have this truncating mutation. There's an early mutation, the rest of this gene is not even there, is not expressed. Right, so when it's really scattered like this, it's usually a sign of loss of function, because it doesn't matter where it appears. Every mutation makes the gene work less well. It could be completely gone as well. That's usually a sign that it's a loss of function mutation, right? It's again, you can imagine if you have a car, you take out parts and you're trying to see when does it make the car run better. It has to a very specific part up. You take that thing, suddenly the car runs better. It's very rare, but most of the time you take parts off, the car doesn't work so well, and that's what you see in this RB case.
Yeah,
okay.
That makes sense.
Okay. All right. So we mentioned that in TCGA studies, a lot of the well-known recurrent mutated genes are discovered. The exception is actually epigenetic regulators, because previously people, when they look at the expression, sometimes EZH2 is an epigenetic factor. It is also highly expressed, but that's not always the case. A lot of these epigenetic regulators, they might only be slightly overexpressed or lower expressed. So differential expression doesn't give you a very strong signal. Also, copy numbers sometimes also don't give you a very strong signal. It's only when you look at the mutations, you see this particular mutation happening. And some of them can happen in, you can see here, the gain of function and loss of function in quite a number of patients, could be as high as like 80% of some patients. And suddenly people say, wow, that's something, right? It's really cool. Especially if you see these, really dark red mutations that are gain of function mutation in specific cancer at say 80% of the chance or the cases, that's when people are very interested in developing a drug that's really specifically inhibit that mutation. Hopefully that can be used to cure the cancer. So about 10 years ago, around the early days of tumor sequencing, many companies started investing in epigenetic drugs, and they're still working on this, and some of them are also already in the clinic. In terms of DNA methylation, you can also see that, for example, with DNA methylation, they see, for example, in this colon cancer type, there's one group of genes that are having very, very different methylation profile in a subset of patients. And they call these a CPG island methylation phenotype. And if you look at so by the way, the columns here are the samples and the rows are the genes, right? And you can see here, this group of samples, their methylation pattern, at least on some of these genes are very, very different. When you look at whether they are genetically different, interestingly, you also see a BRAF mutation. Somehow when the BRAF gene is mutated in colon cancer, these genes, many group of genes, their promoter methylation suddenly change. This is another example for glioblastoma. You can see here, this group of samples, their DNA methylation patterns. By the way, these are from different TCGA studies. Every tumor type, they publish one study. This is in the glioblastoma, they call it glioblastoma CPG island methylation phenotype. There's a subset of patients with very different genes, epigenetic profile. And interestingly, this is associated with another IDH1 mutation. By the way, the scientist who discovered this IDH mutation is a Dana-Farber Bill Kaelin, who won the Nobel Prize last year for this kind of discovery. It's a epigenetics mutation, which is associated with oxygen sensing. And then in gastric cancer, again, you see a subset of these patients. Again, they are associated with viral infection. And if their tumor is caused by the EBV virus, they have very different DNA methylation patterns. And you can see here the EBV infection. And finally, there's in kidney cancer, another group of patients, very different DNA methylation patterns. I guess they still don't. Also, this is another mutation with a gene. So you can see sometimes a particular mutation on the gene is associated with an epigenetic pattern that you see. Yeah, so you can see here these two from both the colon cancer and gastric cancer, the orange group, not only they have mutations or viral infection, they could also have macrosatellite instability. These are repeats in the genome that suddenly become activated. And you will see associated epigenetic changes and also associated, you can actually see this also from gene expression as well. And for the other group, in both IDH mutation and fumarate hydrolase, this mutation is related to the energy metabolism. And in both cases, they have very different survival. So for example, with IDH mutation, yes, it happens, but when the patient has this mutation, they respond better to treatment. Whereas for kidney cancer, when they have this particular mutation, the epigenetic status, different metabolism, they have much worse survival. This is because in general, I think kidney cancer have better survival, and generally glioblastoma has very different a very, very worse survival. And so when they are caused by the same or similar mechanism, one is actually better, the other is worse. That's compared to the norm of those cancer types. Okay, so you can see epigenetic mutations happens a lot, and also they could be associated with other mutations. It could be associated with metabolism or associated with survival. And that's why there are more drugs developed against those. There are also drugs targeting DNA methylation to make the tumors less DNA methylated. And we'll show you some of that in the cancer immunology lecture.